Thank you, Mitch, for the introduction. And before I start, I have to disclose that I do own a company which does the glycan profiling, and we do have a number of patents, and I'm personally inventor on some of them. So part of my talk is advertisement. I will try to make a note when it is. So we all know that efficient patient stratification is the holy grail of precision medicine. And we all try to use genes to stratify patients. The only problem with genes is they do not define everything. And the best example are identical twins. On these photos, we have two pairs of identical twins. Every single letter in their genomes is identical, but their life fates are very different. And this is not defined by the genes. And actually, majority of complex diseases have heritability well below 50%. So there are other things besides genes which should be used for personalized medicine. And one of them are glycans. Practically every protein which has been invented after the appearance of multicellular life is glycosylated. It contains of a polypeptide and a glycan part. And unfortunately, the big majority of mainstream science just ignores the glycans. And the best analogy I can make is that you can study people by researching these two fantastic individuals, Jen Bricker and Nick Vujicic, who were born without their limbs. So you can do a lot of research, see what they can do, study them, and if you study Nick, you can see that he's excellent in typing emails with his big toe, probably faster than me. And then you can do research trying to make a better keyboard or find other ways how to do it. But still, this would not be applicable to the majority of people who use their fingers to do emails. And you may think I'm over-exaggerating, but I'm not. There is a rituximab, a fantastic drug, 20 years on the market, anti-CD20 monoclonal antibody which depletes the B lymphocytes. The only problem with this drug is that its glycans contain this tiny little core fucose. And the core fucose is a safety switch which prevents IgG from killing the cell. So how do you solve a problem if you have a drug with a safety switch which is preventing it from being active? You add more drugs. So we add huge amount of this monoclonals to a person. And then 15 years later, somebody thinks, wait, what if we change the glycan? What if we remove this core fucose? And then they developed rituximab, which is exactly the same drug, anti-CD20 monoclonal antibody, but does not have this core fucose. And then its therapeutic dose is between 100 and 500 times lower. So in theory, this drug could be 500 times cheaper just because somebody looked at glycans and included glycans in what they do. So my advice here is include glycans in your studies. This is advertisement. I sell the test. And you will see much more about the biology. So back to science. Why don't you do glycans? First, it's complicated. Second, it's not encoded in the genome. Actually, majority of protein structure nowadays is just translated from the DNA. We just sequence DNA because it's so easy. We have technologies which are wow, like the talk before me. So it's easy to do. Glycans do not have a genetic template. While every protein is encoded by its corresponding gene, glycans have some kind of complicated networks between genes and the glycans. And through a series of GWAS papers we did in the past decade, we found that it's actually a very large network. And that actually glycoproteins are chemical structures which are being inherited as a complex trait. So it's a completely different paradigm in biology because we don't have a single gene, single trait. We have a complex trait, many genes, encoding a chemical structure. And recently, in collaboration with Grant Morahan from University of Western Australia, we analyzed over 100 strains of collaborative cross mice and showed that in 3 generations of interbreeding, these mice became really different. Only because they reshuffled the alleles which code for glycans. And actually, we see a rapid evolution of mice through alternative glycosylation. And it's not only mice, it's also humans. When you take several random individuals and compare their IgG glycans, and different color is just a different structure, these guys are different. And each of these differences should be treated in the same way as coding mutations. Because a different glycan is a different chemical structure, like a different amino acid is a different chemical structure. So we are seeing myriad of coding mutations and coded and alternative glycosylation. And this is still partly heritable, between 30 and 80% depending on the glycan structure, and partly environmental, both epigenetic, past environment, and current environment. And actually, we don't know so much about it because we haven't studied sufficient number of people. But in my lab, we studied already over 100,000 people. So we do have quite some data about the glycans. And we did not study any people. We studied best phenotype, genotype cohorts we could find. So in addition to glycan data, we have many genetic, epigenetic, whole genome, metabolomic, microbiome data. And actually, we already finished this first big fishing expedition. We have a huge pile of fish, which for us is these Excel sheets. And now we are mining the gold out of those data sets. And actually, this works quite fine. In slightly over a year, we have 10 papers with impact factor of over 12, which is quite decent. And for us, the low-hanging fruits is genetics, because everybody does it. It's so easy. So the first things we did is, let's look for the genes which regulate glycosylation. And our first GWAS, which was published nearly 10 years ago, identified HNF1 alpha, transcription factor, to be the master regulator of antennary fucosylation. And interestingly, the same gene, if you have a single defective copy, you will develop diabetes before the age of 45. Meaning, this is a monogenic, dominant diabetes, so-called MODY, maturity-onset diabetes of the young. And it's mostly undiagnosed, even though the therapy is different, so it would be great if you know if you have MODY or type 2. And we said, okay, if HNF1 alpha is causing MODY, and HNF1 alpha is master regulator of fucosylation, can we use a fucose as a biomarker to identify this subgroup of people with a mutation in HNF1 alpha? We tested it, biomarker potential was quite fine. We went to the prospective clinical trial together with Mark McCarthy in Oxford, 1,000 people, analyzed glycans, sequenced everybody, and when we compared glycans to all the mutations we found, it was lousy. But, as you know, not all mutations are causative of a disease. So when we looked only at the causative mutations, wow, it was a good biomarker. So another aspect of glycans is they can actually be a functional readout for a mutation. Because you have a novel mutation, you don't know what does it do. Check what it does to glycans and then learn, wow, this one is causative. But for glycans, it's more complicated because it's not only genes. It's only epigenetic regulation of the same gene. Because the SNP can deactivate the gene, but also the epigenetic silencing can deactivate the same gene and have the same effect on a glycan. So we ask ourselves a question, is there epigenetic HNF anomaly? Do some people who have type 2 diabetes actually have epigenetically silenced HNF1 alpha? Here we teamed up with Vlatko Zoldoš from the University of Zagreb and first just compared the cohorts. So are people with a different methylation of HNF1 alpha, do they have a different glycosylation? We did see association, but it's association, so is it causative? Then Vlatko developed the tool which can change the methylation of a specific region to check for functionality. I will not go into details here because she has a talk tomorrow, so come and listen to her talk. I will just say that we then moved to our cohorts and found that in a group of type 2 diabetics, and this was done on several thousand people, there is some information about glycans which is indicative of diabetes, but interestingly, also before people develop diabetes. 5 to 10 years before they develop any other symptoms, their glycans are already different. So we went to the large prospective study. There is an EPIC study in Europe. We went to the Potsdam cohort. They have 27,000 people collected over 20 years ago with a clinical follow-up after that. There was 700 cases of incident type 2 diabetes. And then when we looked into glycans at the baseline, they were as predictive for type 2 diabetes as everything else combined. So so-called German type 2 diabetes risk score, glycans are equally good. When they combine them, it's even better. So glycans are predictive of future development of diabetes. But diabetes is not a single disease. There are different molecular pathways to diabetes. And it seems that glycans are associated with one of them, and that one is that if you're in acute disease, if you develop hyperglycemia, so if you put person under stress, if the glucose controls falls out, this is indicated by the glycans. So glycans are practically perfect predictors of this hyperglycemia in acute disease, which is known risk factor for diabetes. And the question is, can we change that? So currently, together with Professor Wei Wang, we are trying to set up a big trial in China where people will be initiated to do lifestyle intervention, stratified based on the glycan risk to see whether we can actually prevent diabetes by early intervention into glycans. So we have this diab-risk test, which is commercial again, for prediction of diabetes. It seems to detect a subgroup of patients which have altered glycans, which are on the path to diabetes. And there are two possible ways currently known. One is something suggested by Jamie Mart and his team, published in Nature Medicine nearly 10 years ago, where HNF1-alpha is on the road of controlling GLUT transporter glycosylation and the half-life on the membrane. The other option is something suggested by Phil Scholl and Chieko Mineo from UT Southwestern, where they showed actually that hyposialylated IgG is actually causing diabetes in the mouse models. So we're currently following that track. Regarding IgG, it's an extremely interesting molecule. We know a lot about its glycosylation because it's easy to study, it's most abundant glycoprotein, many people studied it. So we know this Fuco story, which I already mentioned. It's a safety switch against ADCC, but there's also this sialylation part, which is actually suppressing inflammation. So your IgG, if it's properly sialylated, it's actually suppressing inflammation, not activating inflammation. What happens when there is no sialic acid or no galactose here? So we did a series of GWASs, and we learned there is approximately 30 genes which regulate IgG glycosylation. So there is a network of 30 genes which decides what kind of glycan will be put on your IgG and what kind of functional properties will it eventually have. And the question here was, okay, if this is important, what happens if there is something wrong with this regulatory network? So we did mapping of the pleiotropy. So we checked which of these genes are actually disease-causing genes. And we identified 94 different phenotypes at the SNP level. So the letter in a genome, which regulates IgG glycosylation, is a risk factor for Crohn's disease, type 1 diabetes, lupus, Parkinson, cholesterol levels, dementia. So I'm not claiming that all these SNPs act through IgG glycosylation, but out of 94 of them, at least some of them actually have glycans as an effector of the disease risk, which is genetic. Because remember, alternative glycosylation, putting different glycan on the same site, can have equal functional consequences as a coding mutation. And for IgG, we recently published a large review paper, over 300 references citing what goes wrong with the glycans in the different diseases. And the key question we want to know is, are glycan changes a cause or a consequence of a disease? So what comes first? And this is not easy to study because you have to have historical samples and then a clinical follow-up. But there are some more and more papers coming out showing that actually glycans are either changed initially, so they're a risk factor, or they change much earlier than any other disease symptoms. For example, again, in a collaboration with Wei Wang, we showed that if glycans are changed in hypertension, so people with hypertension have definitely changed glycans, but glycans also change in pre-hypertension. So they seem to change first, and then they drive hypertension. In collaboration with Tim Spector and Jim Wilson, we analyzed over 5,000 people and realized that IgG glycan composition strongly associates with the cardiovascular risk score. So your glycans actually predict your risk of cardiovascular diseases. So this study was correlative. So still we can't say anything about causation, but then we went again to this EPIC cohort, And out of those 27,000 people, we had 500 cases of incident cardiovascular disease. So people who, after they were sampled, they got heart attack or stroke. And again, glycans were highly predictive. So glycans measured today are predictive of heart attack and stroke in the future. And actually, in females, only a single glycan combined with age is more predictive than the AHA score, which is currently the main predictor for the cardiovascular diseases. So it does seem that if IgG glycans are changed, they do something to move you into the direction of cardiovascular diseases. Then we went into the world, asked all our friends, can you send us a couple hundred people from your region? And we collected 27 populations. When we cleared everything for age and so on, we had 100 individuals from each population. As wide as possible age span, we analyzed IgG glycans and learned they're on a line with expected lifespan, but also with the Human Development Index. So depending on where you live, your lifespan will be, expected lifespan will be shorter or longer, depending on the living conditions. And glycans follow that. So if you live in Uganda, your glycans will move toward your late age much faster than you live in Western Europe, for example. And based on all of that, and now this is commercial again, we developed the Glycan age test, which is a test which can predict your biological age based on over 60,000 people we analyzed. And we actually do have it on the market already. St. Catherine Hospital is selling it. We have pharmacists in Croatia selling it. We also have some sales in London, in Beijing. And the idea is that this chronic inflammation is one of the main drivers of aging. And it's also one of the main drivers of many different diseases. And it goes from some kind of a homeostatic condition toward severe disease phenotype. And in the early stage, you can actually revert it by lifestyle intervention. So you change your lifestyle and you do not, your glycans do not become so pro-inflammatory. In the later stages, early therapeutic intervention could also revert the path. But when you come to the severe phenotype, then it's very hard to treat the cause. And actually, in most of the diseases we have, we actually treat symptoms. We are not treating causes. So the question is, how can we change our glycans? We don't have sufficient data. We don't have many people who did a lifestyle intervention, and then we followed glycans for a long time. I'm one of these guys who was followed. I'm following myself for over 4 years. And my glycans go to pro-inflammatory direction most of the time. Beside one little exception here. So what happened here? I took a long holiday, hiked 3-4 hours in the mountains, socialized with the wild goats, and it actually did improve my glycans. They become less pro-inflammatory. But then I had to return back home, work, projects, so on. So my glycan age went through the roof again. So that's me. For me, this is important. For any of you, this might not be valid. You know, every person is different. There is no standard human. But we did a large study on over 800 people in Australian cohort, and we did learn that it's actually central adiposity, which does seem to be the main driver in the majority of people. We did some small studies on bariatric surgery, and we had a patient who, after bariatric surgery, decreased in glycan age for over 30 years and 6 months. So these things are changeable. We had a cohort of elderly people exercising and on a diet for 12 weeks. Their glycans did improve. But then additional cohort we did together with Marcus, and he studied ladies preparing for the bikini fitness competition, which does seem to be a very rigorous sport, and they were actually worse. So too much exercise does not do you good. And the key question, is, what is optimal? And we still don't know. So I'm extremely interested in anybody doing intervention studies on people. Store a few drops of blood. I'll be more than happy to look into glycans. And 10 seconds before my talk is over, I want to acknowledge that this is the work of great team. I have fantastic young people. We work with many of them I worked for 10 years, and they did all the work. I just do the talking. We are extremely collaborative. We collaborate with dozens of institutions all around the world, and we always look for new collaborations because we analyze 30,000 glycan samples per year. We can easily accommodate any interesting study and contribute glycan data to interesting studies, and you do your magic and analyze it together with the glycans and have more information. And of course, to be able to do it, we need a lot of funding. We burn a lot of money on glycan analysis. We have multiple EU grants. We have some European structural fund grants, and I thank them for their support, and I thank you for listening.